Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience-based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the Deep Dive. Today, we're looking at something fundamental, something we thought we understood about how cells handle stress. And it turns out, well, maybe we didn't.
That's right. We're talking about lysosomes, the cell's recycling centers. And for years, when scientists saw them swelling up, vacuolating, especially in diseases like Alzheimer's.
Where you see these images, these bloated lysosomes, and you think, okay, that cell's done for. Catastrophic failure.
Exactly. It looked like the system was just breaking down, accumulating junk.
Right.
But this paper we're digging into today, it completely flips that idea on its head. It suggests this swelling isn't passive failure at all.
Okay, hang on. It's not failure. Then what is it?
It's a defense mechanism, an active, controlled process. The cell is intentionally inflating these lysosomes.
Intentionally inflating them. Why on earth would it do that?
To avoid bursting. See, lysosomes handle degradation, ion balance, volume. It's a tough job. Sometimes things go wrong. Solutes build up inside. Think of it like way too much salt dissolving in there.
Okay, so water rushes in. Osmosis.
Precisely. Water rushes in, the pressure builds, the membrane gets stretched super tight. If it can't expand somehow, it'll rupture, pop, and spill all those digestive enzymes, hydrolases everywhere.
Which is bad news for the cell, like instantly fatal.
Totally fatal. So the cell needs a way to manage this osmotic crisis, this tension. And this paper identifies the manager, a protein called LYVAC.
LYVA? I thought it was called PDZD8.
It was, but the authors recognized its specific role here was so important, they renamed it the L-lysosomal vacuolator. LYVAC, it's the key player coordinating this emergency expansion.
Okay, LYVAC, the vacuolator. How did they even find this thing? Was it just a lucky guess?
Not at all. That's the cool part. They used a technique called turboid proteomics. Basically, they stressed the cells out to deliberately cause the swelling.
How did they stress them? What was the trigger?
They used a drug, apilimod, which inhibits a key enzyme called PIK5. Inhibiting PIK5 is known to cause lysosomes to swell because it messes up how they process and transport stuff internally.
Ah, okay. So PIK5 inhibition creates that solute buildup, the osmotic stress we talked about.
Exactly. It's a reliable way to trigger the swelling. So they triggered it and then used turboid to sort of spray paint labels on all the proteins that showed up right there on the lysosome surface during this stress.
Like putting out molecular wanted posters at the scene of the crime, or, well, the scene of the defense in this case.
Kind of, yeah. And guess which protein was right at the top of the most wanted list?
Let me guess. LYVAC.
Bingo. PDZD8. Now, LYVAC, and what's known about LYBs, or PDZD8, is that it's anchored in the membrane of another organelle, the endoplasmic reticulum, the ER.
The ER. That's like the cell's factory, right?
Yeah.
Especially for making membranes lipids.
That's the one. So the hypothesis immediately jumped out. Maybe LYVE forms a bridge, a connection between the ER factory and this stressed lysosome.
A bridge to deliver materials, like lipids, to allow the lysosome membrane to expand.
That was the idea. A lipid pipeline, essentially, from the ER to the lysosome, mediated by LYVC, to let it inflate safely.
Okay, great hypothesis. How did they prove LYVAC was actually doing this and that it was essential?
This is where figure 1 comes in, and the evidence is, well, really stark. They just got rid of LYVAC, genetically deleted it, made LYVAC knockout cells, or LYVAC-CK.
Okay, so take out the suspected bridge builder.
And see if the bridge still gets built. Right, so they hit these LYVAC-KO cells with the same PIK5 inhibitor stress, or even genetically deleted PIK5 itself.
And the result? Did the lysosome still swell up?
Not at all. Complete protection. The vacuolation just stopped. The cells couldn't swell up like the normal ones did.
Wow. So no LYVAD, no swelling. It really is essential for this specific inflation process, at least under this PIK5 stress.
Absolutely essential. It proved LYVAC isn't just hanging around. It's actively orchestrating that expansion. Without it, the lysosome membrane stays rigid, facing that dangerous tension alone.
Okay, so LYVAC is the hero when PIK5 fails. But is that its only job? Is it just a specialist for that one type of problem?
That's what they tested next, moving into the figure 2 data. They wondered, is LYVAC a general osmotic stress sensor or just a PIK5 specialist?
So they threw other kinds of stress at it.
Exactly.
Yeah.
A whole bunch of different things that cause osmotic issues in lysosomes but are totally unrelated to PIK5. Things like weak-based drugs,
Yeah.
some chemotherapy agents work this way, like metoclopramide.
Okay, drugs. What else?
They loaded the lysosomes up with sucrose, a sugar, to mimic conditions you see in lysosomal storage disorders where stuff accumulates because it can't be broken down.
Right, another way to get too much solute inside.
Yep. And they even just put the cells in hypotonic media, basically. Diluted water outside the cell, which also forces water into the lysosome. Simple osmotic shock.
So a drug stress, a storage stress, and a basic physical stress. What happened in the LYVAC knockout cells under these conditions?
Same result, across the board. In every single case, deleting LYVAC strongly suppressed the vacuolation, the swelling.
So it is a general responder. It doesn't matter why the lysosome is under osmotic strain. Could be PIK5, drugs, storage defects, whatever. If there's pressure, LYVAC steps in to manage the swelling response.
That's what it looks like. It's a master switch for osmotic stress-induced vacuolation.
That really changes the picture.
Yeah.
But how specific is this swelling response? Does the cell use LYVAC for other kinds of membrane problems? Like, what if the lysosome membrane gets physically damaged, like a tear?
Great question. And they tested that directly. They compared LYVAC's response to another protein they know deals with membrane issues called ATG2.
ATG2, what does that one do?
ATG2 is also a lipid transfer protein, but it's known to be recruited when the lysosome membrane is physically damaged, like poked with a hole by a specific compound they used called eloman. Think of it as needing a patch job.
Okay, so LYVA for swelling due to internal pressure, ATG2 for patching physical holes. Did they respond differently?
Completely differently. LYVAC only showed up when there was osmotic stress. ATG2 only showed up when there was physical damage from LMA. They didn't cross over.
That's a clean separation. It really highlights that the cell sees these as distinct problems with distinct solutions. Vacuolation via LYVAC is one strategy-controlled expansion. Membrane repair via ATG2 is another, patching the damage.
Exactly. Two separate toolkits for lysosomal integrity, and it confirms that vacuolation isn't just, damage happening. It's a specific, regulated program run by LYVAC.
Which brings us to the nuts and bolts. How does LYVAC stuck on the ER actually inflate the lysosome? You mentioned a lipid pipeline. Let's break that down. They described it in 3 steps, right?
Yeah.
Signaling, recruitment, transfer.
Yep. Step 1 is the signal. The stress lysosome basically sends up a flare. When osmotic stress hits, an enzyme on the lysosome called PI4K2A gets activated.
PI4K2A. Okay, what does it do?
It generates a specific lipid signal molecule called PI4P right there on the lysosome surface. This PI4P acts like a flag, recruiting other proteins.
Which proteins?
A group called ORP proteins. These are known lipid exchangers. They get recruited by the PI4P, and they start the process by trafficking 2 specific lipids from the ER to the lysosome: phosphatidylserine, or PS, and cholesterol.
Wait, so before LYVAC even gets involved, these ORP proteins have to sort of prepare the landing site on the lysosome with specific lipids, PS, and cholesterol.
Exactly. It's like they're laying down the welcome mat. This influx of PS and cholesterol creates a unique lipid signature on the stressed lysosome surface. That signature is crucial for step 2, recruiting LYVAC itself.
Okay, so the site is prepped. Now, LYVAC needs to get there. How does it know to go to that specific stressed lysosome and not just any lysosome? You said it uses like a triple check system.
Right. It's very specific. LYVF has 3 key parts that help it tether correctly. First, it's already anchored in the ER membrane. That's stable. Second, it has a domain that specifically binds to RAB7.
And RAB7 is a marker protein found on lysosomes, right?
On late endosomes and lysosomes, yes. So that ensures it's targeting the right kind of organelle. And third, LYVAC has another domain, a C1 domain, that specifically recognizes and binds to that phosphatidylserine, the PS lipid, that the ORP proteins just delivered.
Ah, so it needs the ER anchor, the RAB7 lysosome signal, and the PS stress prep signal. All 3 have to be there for it to lock on.
Precisely. Its multivalent binding ensures LYVAC only forms that bridge at the exact time and place it's needed, an osmotically stressed RAB7 positive lysosome that's been prepped with PS and cholesterol. Highly controlled recruitment.
Okay, LYVAC is recruited, it's tethered, bridging the ER and the stress lysosome. Now for step 3, the actual transfer. How do the lipids move across? LYVAC has that SMP domain, which forms the channel or bridge,
Right.
but what pushes or pulls the lipids through?
This is where the physics gets really interesting. They found two main driving forces. First, there's a chemical potential gradient. Well, think about the ER membrane. It's relatively neutral, maybe a bit crowded. The lysosome membrane, now thanks to the ORPs, is enriched with PS and cholesterol, making it chemically distinct, maybe more charged or ordered. Lipids generally prefer to move towards areas where they fit better or where there's a stronger chemical attraction. It's energetically favorable to move from the generic ER to the specialized lysosome surface.
Okay, so chemistry helps pull them over. What's the second force?
This one is maybe even more powerful. The osmotic membrane tension itself, remember all that water rushing in, it creates high physical tension on the lysosome membrane, stretching it taut.
Right, the very problem LYVAC is trying to solve.
Exactly. And that tension acts like a vacuum cleaner, effectively pulling lipids across the LYVAC bridge from the ER. They actually showed this in lab experiments in vitro. The more tension they put on the receiving membrane, the faster the lipids transferred through LYVAC.
Wow, so the stress, the tension, is actually providing the driving force for its own relief mechanism. That's incredibly elegant.
It really is. The crisis fuels the solution.
And they could actually see this lipid movement happening in living cells. How?
Yes, using a technique called stimulated Raman scattering imaging, or SRS. It's label-free, which is important. You don't want fluorescent tags potentially messing things up. SRS lets you directly visualize the chemical bonds of the lipids themselves.
And what did the SRS imaging show?
It showed exactly what the model predicted. In stressed cells, the lipid concentration inside the lysosomes went up. And crucially, they could see a corresponding decrease in lipids in the bits of ER right next to those lysosomes.
A depletion zone in the ER. Clear proof of lipids moving from the ER to the lysosome on a large scale.
Undeniable. Directional large-scale lipid movement driven by osmotic stress mediated by LYVAC.
This whole mechanism just elevates the ER lysosome connection, doesn't it?
Yeah.
It's not just a passive supply line, it's an active dynamic stability control system. It feels like a major shift from how we usually think about lysosome regulation, which often focuses on ion channels or master gene regulators like TFFEV.
Absolutely. This introduces a whole new layer, a fundamental mechanical countermeasure, volume control through lipid transfer, orchestrated by LYVAC. It puts these ER lysosome contact sites center stage as critical governors of organelle stability.
And the implications. They seem huge. Where does this lead us?
Well, first, think about those neurodegenerative diseases we mentioned, like prion diseases, where PIK5 function is often impaired. This LYVAC mechanism, while protective initially, could be a double-edged sword.
How so?
Chronic excessive vacuolation driven by LYVAC in those disease states might actually become detrimental over time. Maybe it disrupts normal lysosome function in other ways or makes the cell fragile. So perhaps modulating LYVAC activity, maybe inhibiting it slightly in those chronic conditions, could be a therapeutic strategy.
Tuning the response, that makes sense. What about other areas like cancer?
That's another really striking finding. Remember those weak-base chemotherapy drugs? They often get trapped, sequestered inside lysosomes.
Right, the cell tries to hide them away.
Well, the study showed that when they knocked out LYVAC in cancer cells, those cells became more sensitive to a drug like doxorubicin.
More sensitive. So stopping the swelling helps the chemo work better. Why?
Because if the lysosome swells up, massively vacuolates, it effectively dilutes the concentration of the drug that's trapped inside. The drug becomes less potent. But if you prevent that swelling by knocking out LYVAC, the drug stays concentrated in a smaller volume.
Making it more effective at killing the cancer cell.
Exactly. So this suggests a potential therapeutic angle. Maybe combining existing chemotherapy drugs with a LYVAC inhibitor could overcome some forms of chemo resistance, boost the effectiveness.
Fascinating. So we've gone from seeing swollen lysosomes as just a sign of death to understanding this intricate adaptive dance involving ER lysosome bridges, specific lipid signals, and physical tension, all orchestrated by LYVAC. It's really beautiful cellular engineering.
It truly is. And it repositions vacuolation from pathology to physiology, a programmed response.
So what's the final thought you want to leave our listeners with? Something to chew on?
Well, this paper clearly showed that membrane tension can act as a physical driving force pulling lipids across the LYVAC bridge. It makes you wonder, if mechanical strain is such a powerful signal for lipid transfer at ER lysosome contacts, where else might it be playing a role? What other critical cellular decisions, maybe beyond just basic survival and rupture avoidance, are being influenced or even controlled by cells actively sensing and responding to the mechanical forces on their internal membranes? That's the next frontier, I think, the mechanics of the cell.
The secret life of organelle mechanics. A fantastic place to end. Thanks for that deep dive.
My pleasure.